today we're going to look at a couple of case studies. So I'll present one for 30-40 minutes this morning. It's related to some recent work that I've been doing. Andrew and/or one of his current students is going to talk about some of the work they've been doing, and there'll be a recorded case study from our Hobart tutor, Jawahar Patil. So they'll all be available on the Milo site. Andrew's and mine probably by tomorrow morning. Jawahar's won't probably pop up till Friday. He doesn't give his tutorial till Thursday. So the idea of the case studies is to give you a real example of how molecular approaches can be integrated with other things, not only as a sole tool for determining or investigating a particular question or problem. I did have some snazzy music to go along with this, but I just can't get it to work when I open this slider. But anyway, so basically I'm framing this bit of work in the context of DNA profiling to identify a killer, hence the CSI kind of thing. But before we get on to talking about that, I just want to give you a bit of background on the type of organisms I spend a lot of time working on. I don't just work on bacteria as we've done this year. I do work on other microbes, bacteria, particularly I'm interested in the interactions between phytoplankton cells, microalgae, and the bacteria that live on or around them. The other area that I tend to spend put a lot of focus on is harmful and toxic algae. So if we quickly look at microalgae and phytoplankton, this is a scanning electron micrograph of a sample taken, actually this is in the southeast in the Derwent River. You can see quite a few complex looking cells in there. Most of the ones in this sample are what we know as dinoflagellates, the group that particularly interests me. But you can see these long, skinny bits and pieces are tubular shapes. They're actually diatoms. So between the two of them, diatoms and dinoflagellates make up the vast majority of biomass, if not the vast majority of species in coastal waters. So they're particularly well adapted to, more nutrient-rich coastal situation. They're small, so phytoplankton range from a couple of microns, so in a similar range to the larger bacteria, all the way up to a couple of hundred microns, and there are a few which are bigger, perhaps almost up to a millimetre in length. They're a different thing to seaweeds in one sense, in that seaweeds are multicellular, but in fact if we look at the phylogeny, the relationships between the organisms, particularly using molecular approaches, what we see is that red, brown and green algae are actually just what we might call multicellular protists that fit in amongst all of these microalgal and protistan groups. So classically thinking about things as being plants, algae, protists, fungi and animals just doesn't work. There's probably, I think last count, maybe 40 or 50 different phyla, not just 4 or 5, or you could even call them kingdoms. So red, brown and green algae are actually just offshoots of various groups within all of the protists. That's what diatoms look like in real life. So really pretty organisms that are contained in silica shells called frustules. They've got a lot of morphological diversity, and those solid structures actually allow a fair amount of information from which to characterise and identify and distinguish different species. So really kind of information-rich morphology, so we can get a long way without needing any other techniques like molecular biology. But as we start looking at these groups with molecular biology, what we find is our assumptions based on morphological similarity and difference actually are not overly accurate in a lot of groups. We find that, yes, there are some similarities at around the genus level, but once you move beyond the genus level, there's a lot of convergent and divergent evolution that tends to confuse our view if we base our assumptions on morphology. This is a group that I'll be talking about most of the rest of the lecture, dinoflagellates, a few different examples there from some of my past work. This one particularly here, chain forming one on the side, I've spent a lot of time working on. I think when I first finished my undergraduate, I went to work with somebody working on that species, and I'm still working on it 30 odd years later. So there's a mixture of light micrographs as well as scanning electron micrographs. Most of you probably don't realise that you see dinoflagellates quite often. Perhaps in this, in the example of one called Noctiluca, that is one of the primary causes of bioluminescence. That's been particularly common around the east and southeastern coasts of Tasmania in the last several years. So when you see these at night, these bright flashes of blue or waves that are crashing with this blue, it's the dinoflagellates that are causing it. And basically the thought process behind why these organisms produce bioluminescence is because they think it might be an escape mechanism. So predators which are feeding on them can be temporarily confused by a brief flash of light. And when you get an accumulation of the organisms all together, you actually have enough light being released as the animals are disturbed to release enough light to be able to see it quite clearly on a moonless night.
It's not toxic itself, is it?
No.
Like it can drain all the nutrients out of the area, but it's not.
Yep, so Noctiluca, not particularly, doesn't produce a toxin per se, but it's a large, it's a predatory type feeder, and it has a large sac associated with it, so it's actually quite large and visible. It has a large sac which it contains a lot of nasty things in it, which are things like ammonia and so on. And so when you get a large bloom, and then you get the bloom collapsing and lysing, you do get release of things like ammonia into the water, which can upset other marine organisms. But it doesn't produce a toxin classically, or a secondary metabolite toxin, like a lot of the other dinoflagellates do. So quite a lot of dinoflagellates, perhaps a few hundred different species, we now know produce a range of different products that are directly toxic in one form or another. Back in 2012, there was a large outbreak of mussel or paralytic shellfish toxins in mussels on the east coast, and that resulted in a fair range of news coverage. Mussels from spring-based seafoods had been exported to Japan and detected during the routine screening for these kinds of compounds when they entered Japan. So that caused a huge problem, not only for the mussel industry, but also seafood industry generally. So you can see a few different things. 2012, even made international news at various stages. Scallops, so it ended up in mussels, oysters, scallops, lobsters. Basically anything, or basically ended up going from the pelagic food chain also into the benthic food chain and into things like lobsters that feed on benthic invertebrates. So toward the end, or just after the bloom finished, we started to see the toxicity turning up in other benthic organisms, things like abalone and rock lobster. Gosh, I didn't realise I had quite so many. All right. So that particular event affected seafood and shellfish from the area around Marion Bay here in the southeast, on the eastern side of the peninsula, all the way up to just around the northeast of the coast. So this is where the toxins were detected, not so much the causative organism itself. Also in the southeast, there was toxicity occurring at the same time due to a different organism. That chain-forming organism I showed you a few slides back regularly produces problems in this area and has done since the mid-1980s. Just to tell you a little bit about paralytic shellfish poisoning, and this is true of a lot of shellfish toxins. The toxins are produced by phytoplankton, particular phytoplankton species in the water column. They are then fed upon by bivalves or other filter-feeding organisms. And then human consumers can eat the intoxicated bivalves, and then they suffer paralytic shellfish poisoning. The toxin itself is a tetrahydropurine, an alkaloid-like structure here. Has a lot of nitrogen in it. It varies considerably in its toxicity to vertebrates and humans. So, like many of the toxins produced by algae as well as things like fungi, there's a lot of variation. So the compound has 1, 2, 3, 4 different sites that vary in terms of the side group, so the chemical group that be on the side, and so there's half a dozen different variants that change not only the conformation of the compound but also how it interacts with the nervous system of humans. So the way paralytic shellfish toxicity works, this particular compound, saxitoxin, depolarises axonal membranes in our nervous system, which basically shuts down our autonomic nervous system, and the symptoms that tend to happen are that your breathing starts to stop, or stops. So in severe cases of poisoning, the only solution is to put somebody on a respirator and wait for a couple of days until the toxin is excreted from the system. We have really only had a few poisonings in Tasmania, some back in the 80s, when this particular organism, Gymnodinium catenatum, turned up in the southeast, and we had a couple of poisonings in 2016 from wild collected shellfish at Little Swanport, which is due to the organism I'm going to be talking about today. So these different varieties and versions of the toxin can make a huge difference, 10, 100, 1,000-fold difference in the toxicity to humans, even though you might have the same amount of the compound there. Basically, that's because these different side groups affect the way the compound interacts with the axonal membrane. So basically stops conduction of nervous signals. This is what Gymnodinium catenatum looks like. This is the organism I first started working on. Has these fantastic long chains, really easy to identify. There's very few other species that look like it, or at least so we used to think, but that's not quite so true anymore. So generally, when you see that particular organism, it's really obvious which species you've got, and we know that this one produces a toxin. On the east coast of Tasmania, it's a different organism involved. At the time, it was thought to be this organism, Alexandrium tamarinense. So, similar size to Gymnodinium catenatum here, but it differs in the sense that Gymnodinium catenatum doesn't have a hard shell around the outside called a theca. Alexandrium, you can see these little refractive structures around the outside of the cell. There's a hard shell called a theca around many dinoflagellates. If we actually look a little bit at this shell, it has little sutures and plates that are joined together in complex patterns, and it's the shape and pattern and structure and the way they join together that give us information that we can use to identify the organism. So based on morphology, if you know what you're looking for, you can tell by looking at either the top or the bottom half what kind of genus it is and what kind of organism it is, maybe even down to which species. Here's a different image, same organism, and it's the details and positions and relationships among all these little plates that all have little special names for those of us who do taxonomy on these organisms. So depending on how they're positioned, the shape, where various holes and things are, we can usually identify species pretty easily. And so this was the organism that was in the water column at the time that the toxins appeared on the east coast. Unfortunately, the monitoring program missed this organism for a couple of reasons. Partly because it happened in September, October, and the east coast, and particularly during the winter and spring, had been classified over the years as a low risk area, so there wasn't a lot of sampling going on of the shellfish. So this organism really hadn't appeared and been a problem in Tasmania in the past. Unfortunately, the taxonomy and the relationships of these groups are actually quite complicated. So Alexandrium tamarense, that particular organism I've shown you a few images of, occurs all around the globe, where it's associated with shellfish toxicity in a number of areas, particularly northeastern US and also northern Europe and South America. If you look at DNA sequences, and this is DNA sequencing of the large subunit ribosomal RNA, so a very similar gene to the one that we've been working on with Vibrio. Bacteria have a 16S ribosomal RNA gene. They have a 23S, so a small one and a large one. The 16 and 23 refer to the mass of the RNA that it produces. The large subunit RNA in eukaryotes is called the 28S ribosomal RNA gene. Structurally quite similar to the bacterial large subunit, but different enough that we can target it with PCR and amplify it without really needing to ensure we've got bacteria-free cultures and so on. So a number of researchers in the past, including myself, have sought to examine culture and sequence these organisms, see if we can understand the relationships between populations around various areas of the globe. What we've come up with over the past 15 to 20 years is this organism called Alexandrium tamarense, which I showed you. Looks pretty much the same all around the world, but there are 5, and we now know, actually recently have found a 6th different group. So when you actually look at the DNA, they cluster into these discrete patterns on our phylogenetic tree. If we go from the bottom, group 5, as it was called, is actually restricted, we think, to Australasia. So it was one that I first discovered back in the late 80s, early 90s. See, ATBB01 is actually a strain code from down the river here at Bell Bay. That's why it's BB. Doesn't produce any toxins. So we're still working through how toxin production works and the metabolism and genetic machinery involved in toxin production. But if we look at this group of organisms, anything we've isolated that belongs to this group 5, so far, no toxins. Group 4, generally known around the globe as catenella because it forms chains of cells, a bit like Gymnodinium catenatum. Always toxic, always produces a fairly similar suite of those different types of saxitoxin. So quite a common organism, very similar to what we would have called Alexandrium tamarense. The only morphological difference is whether it forms the chains or whether it doesn't. As it turns out, that chain formation is not a great way of determining the relationships among organisms. Group 3, generally in southwest England and western Europe, generally doesn't produce any toxins, and you can see same kind of species name has been applied to everything in that group in the past. As it turns out, the first description of this organism was from the south coast of England back in the 1920s. So if any group is going to be our type organism, the thing that truly should be called Alexandrium tamarense, it's this one. All right? These ones, DNA tells us they're actually quite different. All right? This is quite a few different bases of DNA between those two groups. Group 2, also non-toxic, appears in the Mediterranean and southern Europe, doesn't produce any saxitoxins. And at the top, group 1, you'll see the species names. If you look across that tree, there's a few different ones there: tamarants, fundients, catenella, and so on. That's quite a morphologically variable group, hence all the different names. Some form chains, some don't. Some have a few different holes and pores in plates, and some don't. Yet when we look at them genetically, they all pretty much fall within a clear group up the top of the tree. Alright, so that one there's a bit of a mystery. We're still trying to sort out the differences between morphology and genetics, but what we have discovered, when you look at any one particular population, you actually can't really tell the difference. There are a few minor things that might put them in one particular species or not, but we know that the species don't correlate well to the relationships we've discovered from DNA. So effectively, when you see any particular cell, you're not going to know which of any one of these 5 groups it belongs to unless you do some DNA sequencing.
How many groups did you say there is there? There are 3.
5 all up. So 5, 4, 3, 2, 1.
Why are those 2 on the bottom not separate groups?
Those ones?
Yeah, and the ones at the bottom as well.
These ones?
Yes.
Alright, so these belong to species that morphologically we can quite easily distinguish, and genetically they're also distinct. So the reason these are in the tree is actually to provide a route. So remember we talked about routing your tree so that we can orient everything from early to late, or the past and present in terms of evolution, yeah? So all this group is doing is actually making sure we know where the bottom of all of that group is. This is our group of interest. This is our group that we're using to provide the route, to orient the tree in time. All of these species are actually not too dissimilar to that, but they're sufficiently morphologically distinct that there's no problem with distinguishing any of these. We know what these are, and in fact, a couple of them produce toxins, but most of them don't. All right? So this is the group that we really can't tell apart with morphology. And it's complicated. Effectively, the most logical way to deal with this is to actually consider them 5 separate species that we actually can't easily distinguish using morphology. Alright, so if we have a look at group 1, actually no, if we have a look at the whole lot, Alexandrium tamarens complex as it's known, which has these 5 different types in it. Look across the globe, we've got group 1 here, there, up there, here, up in the north. We've got group 4, which is the chain forming variety in southeastern Australia and New Zealand, and also up here in Southeast Asia and Japan and so on. So there are regions where you might find a couple of different types occurring together. Australia is one of those. We had the non-toxic variety that I first isolated and identified on the north coast of Tasmania. We've since seen blooms of it occur at a few spots around the southeastern and southwestern Victoria. Right, so that's what number 4 looks like. So when we say catenella, which is our morphological species, we see these chains of cells that effectively look just like a chain of Alexandrium tamarens cells like any of the others. And if you just see a single cell of those, you don't know which one's which, and morphologically these are not overly different. Not different enough consistently to call them or to be clear what species you're dealing with. So over the years, this particular genus Alexandrium and this species have turned up basically back in the 50s, and then over the years myself and a colleague have picked it up at various places around southeastern Australia. Alexandrium tamarens, so the difference here being that they don't tend to form chains of cells, or at least not long ones, and they have a particular hole in one of the plates on the cell which is either present or absent. Tamarens has a little hole on the plate that you can see. Canonella tends not to. It seems really minor stuff, but when you've got very little morphological information to work with, or differences, you tend to pick up on the things that you can see that are different between organisms. This is what morphological taxonomy is like. You've got to work with what you have. But we know from our genetics that these two organisms actually could belong to a couple of different groups. So morphology won't tell us everything. So over the years, we've discovered different versions here. So either ones where we don't know whether they were toxic or not, ones that were low toxicity, which were probably that group 5, and ones that were toxic. And this one, we have some, or did have some cultures from 1997 that produced toxins and didn't appear to be the chain-forming variety. So we suspect that they may have been Alexandrium tamarense, and a toxic variety, not a non-toxic variety. All right, so in 2012, when this outbreak all occurred, we didn't really know what the source was, or in fact what the identity of the organism was. We knew it was probably an Alexandrium tamarense complex species, so one of those 5 different groups. But anything that looked like Alexandrium tamarense in Australia had so far to this point always been the low or no toxin producing one. So we're a little bit confused. So in late October, early November, we couldn't find any in coastal waters, and the toxicity appeared to be going away slowly. So I took a trip out with the Spring Bay guys out toward the shelf edge in the hope that, well, what I knew about this organism in other parts of the world was that it tended to be associated with the shelf edge and shelf break, and that maybe we were getting large populations out there that were being moved inshore under particular weather conditions. So I went offshore, and interestingly, as we went out toward the shelf edge, so it's 125 metres deep here, there's the surface up there, all the way out, I could see this water column structure. So what looked like zooplankton and or fish feeding at about 40 or 50 metres. When we got out to the shelf edge, which is about just over here on this map, there was a big blob at about 60 metres depth. Usually that kind of mark suggests that there's a large zooplankton population there feeding away on things. That's what's giving us our echo returns on the sounder. So I figured if we sampled down somewhere just above there, we might find the phytoplankton, which would have been what the zooplankton were feeding on. So basically out in the ocean, particularly under relatively calm sea conditions, you get stratification. Organisms don't appear randomly throughout the water column or consistently. They might be at particular depths. Particularly out on the shelf edge, I knew from other areas of the world, this particular organism often was found out at depth on the shelf edge. So we did a bit of sampling, and we were fortunate enough to find a few cells, not a lot, but some, and we managed to culture them. These are all actually taken from the surface, but there are also cells down at the 60 metre mark. Interestingly, all this time we couldn't find anything inshore in the sites where the shellfish were. Did a little bit of basic culturing work to determine what kind of conditions they liked. What's interesting is their optimal temperature is actually quite low. They grow best at around about 13 through to 15 degrees. I had a bit of trouble getting them to grow first up because this species I'd worked with in the past locally, typically I was needing to use temperatures in the low 20s. This particular variety, whatever it was dying at temperatures above 20. So once we lowered the temperature down, we could get them to grow. We also did a little bit of sampling. So when I did that, transect across the shelf, we collected a bunch of samples, and what we found was there were cells in here and in the bay nothing all the way out, and then on the shelf edge was where we found a few more cells. Alright, so this is cells per litre, so this is not exactly very high concentrations. So 60, 20 out here around the shellfish lease, nothing all the way out past the shelf edge, and then the highest concentrations, although not terribly high, a few kilometres above the slope, basically, shelf slope. So this was suggesting that this organism may well have been actually out on the shelf edge a lot of the time rather than in on the shelf or inshore. When we did some DNA sequencing, so very similar to what you guys did with your Vibrio, and we did a bit of comparative alignment with information on the database. And this is basically a graphical representation of the sequences. Where it's grey, the sequences are identical. Where there's a difference, it's colour-coded by which kind of base has been substituted. So we look across these, we can see this is one of my isolates from off Triabunna, compared with those different, or representatives of those different groups I showed you on that tree earlier. So we can see that they're quite a bit different even over this relatively short region of DNA from group 5, group 3, group 4 and group 2, but they're actually most similar to group 1. So this is a strain actually from just north of Alaska, so in the Arctic Circle. This is another one from, I think, Northern Europe, and another one from northeastern USA. You can see, we look at another bit of the sequence, again, lots of differences between all these, but actually very few, except maybe a few inserts and deletions, separating these isolates from Tasmania from all the other group 1 strains I compared it to. This came as a real surprise, actually. I was just heading to a Molluscan Shellfish Safety Conference where I was supposed to be talking about what we'd found, and one of my students had just picked up the sequences that had come back from one of the sequencing services, and I did a quick alignment the night before I did my talk, and my jaw nearly hit the floor. I really didn't expect to find this organism down here, because it had never been detected anywhere in Australasia before. So I guess then the question is this organism, let's go forward a bit. Alright. So if we look at these new sequences, where do they fit? So if we go back to the bottom of the tree again, here I've used just a single out group in my analysis, Alexandria maffini. Very similar organism, but morphologically distinct enough to be pretty clear about what it is. So that's the root of the tree. Here are our groups again, various isolates that I've made in 2012, 2013 in other areas. We can see this is the group 5. So these are the non-toxic variety, which recently have been reclassified as a new species, Alexandrium australiense. So rather than trying to call them all tamarins, those of us who work on the taxonomy of these organisms decided it's far better to actually call them different species names, because functionally that's what they are. The downside is you actually can't tell them apart using morphology, which is a problem for most monitoring programs. Group 4 is now known as Alexandrium pacificum. There's a range, a little cluster there, including isolates from Australia. Group 3 is now Alexandrium tamarense, so that's the one that was first discovered and identified and described from the south coast of England, right, back in the 20s. So this one has, in taxonomy, what we call priority. So if you go back and look at the type material, the description, the diagrams that were in that first description of this particular organism, we can clearly describe organisms that it was Alexandrium tamarense. We can clearly see that at the type location, which is actually near Plymouth, in the Tamar River, Tamar Estuary, hence Tamarense, that should or corresponds to the original type description. So under taxonomic rules, that has to take priority. So that one gets to keep the original name for all of these 5 different things. And we have to generate some new names for all the things that we now know are different, even though they look the same. So that one gets to be called out A. tamarense. Group 2 from Mediterranean has been renamed A. mediterraneum. I didn't come up with these very boring names. And A. fundiense is still the problem one. All right, and that's where the organisms that cause the toxicity problem on the east coast actually belong. This one is a problem not only in terms of taxonomy but also the diversity morphologically. So you get 3 or 4 different morphotypes that occur all within that group, spread pretty much around the globe, this particular organism. It's ended up with a name, Alexandrium fundiense, that already existed, but on the basis that membership or descriptions of that organism were first given the name Alexandrium fundiense in northeastern US, so Bay of Fundy region. All right. So based on taxonomic priority, there's an argument that group should be called Alexandrium fundiense. There's still a bit of an argument going on about taxonomists about priority and type locations, particularly of fundiense and pacificum. It hasn't yet resolved, so we may end up with a few more changes of name in the future. It's a bit hard to tell which way that'll go. But this is the situation at the moment. We now have 5 different species. One of them is A. tamarense. The rest are all related species, and the only way we can pick them apart is using some DNA sequencing. So let's have a look at Alexandrium tamarense group 1. So when I first got the sequences back, the first thing I did was try and plot a map, see if there was any obvious pattern. And this is the distribution of Alexandrium tamarense if we only look at that group 1. So we go through all the records and get rid of all the ones that we think don't belong to that group. So all the other 4 different types where there's reports. So any confirmed report which we can ascribe to group 1, that particular genotype, basically is distributed like that. So that's not worldwide once you deconvolute all the records for 5 different organisms effectively. So that clearly tells me that if we look at that distribution, it's bipolar. It's an organism that generally inhabits colder regions, and if we plot the 15-degree isotherm, which is the average sea surface temperature is 15 degrees, at that point it's less down here and it's more in here. It pretty much conforms to north or south of that average sea surface temperature. So this is the kind of average sea surface temperature that is very close to the boundary of what we call polar or cold temperate and temperate. All right, so you might call it Antarctic associated and Arctic associated versus temperate, which would tend to be in that region, about 15 to 20 degrees average surface temperature. Once you go north or south of these points, you're in tropical, warm temperate tropical. All right, so this is an organism that likes cold water. So based on a bit of work with DNA sequences and a bit of work looking at distributions, we can make some interesting inferences about this that are really useful in terms of understanding shellfish toxicity on the east coast. So one thing is we follow that isotherm, we can see it pretty much hits just the northeastern tip of Tasmania, but if we base our inferences on these kinds of distributions, one thing we can say is we don't expect this organism to be a problem further north. All right? So also the argument, well, maybe this organism has moved south with the strengthening south East Australian current. Right? So over the last 10, 15 years, we've seen a strengthening of the East Australian current, bringing more and more warm water down the east coast of Tasmania. This doesn't look to be the spread of a warm temperate species south with that medium-term climate change. If anything, it's more likely to have actually come from more southerly climes, or it's potentially always been here, and changes in the way the East Australian current are working and how it interacts with the coastal shelf have created an opportunity for this organism to create blooms where it probably hasn't in the past. Certainly any time since I've been working on this problem in the 80s, we've not seen toxicity in shellfish at this scale before 2012. Interestingly, we suspect the 2012 bloom turned up from looking at the toxicity, and we know roughly how long it takes to accumulate and disappear from shellfish. We probably estimate that the bloom probably kicked off in early September along the east coast, and subsequent years have suggested maybe August, September is when it starts to build up. I've actually got old samples that I took in September '89 where we saw quite high concentrations of organisms that look just the same in early September. At the time, we assumed they were the non-toxic variety, the one we now know as Australiense or Group 5, because that's all we knew about Alexandrium tamarense in southeastern Australia at that point. And we made the assumption, possibly incorrectly, that that was a bloom of non-toxic version, now known as Australiense. It could just as easily have been a bloom of Group 1 or Fundiense, and we maybe missed it, and maybe some people could easily have got poisoned if there'd been a substantial bloom. Anyway, unfortunately those samples are not in a condition where we can go back and look at them with molecular methods. They've been in formalin for too long at a too high a concentration for us to be able to really use the DNA. One of the other inferences we can make is my colleagues in New Zealand probably ought to keep an eye out for it. So far, or at least at that time, it hadn't been detected in New Zealand, this particular Group 1 variety or Fundiense. But just looking at the isotherms, would suggest probably the south, the eastern coast and the southwestern area would be areas where you might expect to find it. And my colleagues at Nelson, just in this little area here, tell me a few years ago that they've detected the genotype using quantitative PCR, so that they think it's out there, but so far they haven't found it and been able to culture it and identify it. So the likelihood is it's out there in this central and eastern region of the South Island of New Zealand, but it just has to occur in sufficient concentration for anyone to go out, find it, isolate it and culture it. Alright, but we suspect it's also in this region as well. Okay. So there we are, thrown on the map. So if we look at that, in retrospect, we probably would have expected to find it here at some point. Alright, so this is a distribution that suggests, well, it's probably natural, not some kind of introduction process. We can't rule that out. But if we look at the distribution on the globe, this is where we would expect to find it. Now, the reason why we may not have found it here is it typically is associated with the coastal shelf and shelf edge, so it needs to be near land. All right? There's not a lot of land in the southern hemisphere once you get down to this point. All right? So really, this is the only area left in the southern hemisphere we might have expected to find it. Whereas there's a lot of land and coastal shelf edge in the northern hemisphere in that climatic zone, and we see the organism pretty much spread right across the globe at those northern latitudes. We've done a bit of mapping using both net sampling, isolation and DNA sequencing of what we find in the samples. We have also done some quite a bit of qPCR analysis using primers that distinguish each of these three groups that we know exist in Tasmania. Also using a qPCR that detects the saxitoxin, or one of the saxitoxin synthesis genes. We know the first couple of genes in that synthetic pathway in dinoflagellates. About 2011, 2012, we discovered that, and that's been developed into a qPCR that we can apply to detect the presence of the gene. It doesn't necessarily tell us which organism it's coming from, because the gene's actually quite conserved, but it does tell us whether there's a toxin-producing organism in the sample. So by a combination of quantitative PCR as well as isolating cells and genotyping using the same methods you guys have been using, we've been able to determine that group 1, which we now know as Alexandria fundiense, occurs at relatively low concentrations down here. It's scattered around here and all the way up the east coast. So far, the north coast seems clear of it. And whenever we find Alexandrium tamarense on the north coast, it's always so far been Australiense, group 5, the non-toxic variety. One of the complications for the qPCR for saxitoxin synthase A is that Australiense actually has that gene, but it doesn't produce any toxin. So, if you like, it's a false positive. We don't fully understand yet why it doesn't produce the toxin. In most of the other dinoflagellates we've examined, you have the gene, you produce saxitoxin. So there's an interesting story there in trying to understand why synthesis seems to be down-regulated or switched off in that particular species. I have another organism plotted on this map, also group 4, so this one's known as Pacificum. Really only seen a few times here around the Triabunna Spring Bray region. Haven't really seen it anywhere else. So that's a potential problem. And there's this other organism called Ostenfeldii, quite distinct. We can tell it apart morphologically. Similar size, looks a lot like tamarense, but it has a different arrangement of some of the pores and plates. We can tell that one apart quite easily, and it's quite uncommon, but when it does appear, it produces a bucket load of toxins. It's highly toxic. So we've been mapping that one as well, just so we know where it's likely to be. Okay, so we're getting some sense of how these 3 organisms or 4 organisms are distributed around the state, which allows us to develop more refined risk mapping.
What's the west coast one?
That's a funny colour because we don't know exactly what it is yet. So there was one cell noted by one of the guys at AST who do the plankton monitoring. So they look for cells of concern, but we couldn't go back and verify which species it was. Again, using fixatives, the common fixatives, you've only got maybe a week or 2 to work with the samples before they're no good for DNA. So one of the recommendations we've made for the routine biotoxin monitoring programs, they ought to actually be also taking samples and preserving them in a way that we can actually use them for DNA analysis, which would allow us to go back to older samples when we need to and say, well, hang on, what organism was that? Because we can't tell them apart by morphology. We have to use DNA or quantitative PCR analysis to be able to pick which one is there. We've done a reasonable amount of qPCR analysis over the last few years, funded by the Fishery Research and Development Corporation. And what we find in any particular bloom is that on the east coast it tends to be dominated by fundiense, right, the one that we thought was responsible for the 2012 event. But depending on where it is, what time of year, we often find an equal mixture of group 4, Alexandrium Pacific, the chain-forming one, and group 1, fundiense, the non-chain forming variety. Interestingly, every culture I've isolated over the last 4 years or so is always group 1, and we don't see any chain-forming cells in the water column and the fixed samples. So we're not really sure what's going on there, whether we're getting a bloom that's a mixture or even whether or not some of the group 1 isolates actually may have different classes of sequence. Some that are more like Alexandrium fundiense and other sequences in there that are much more like Alexandrium sificum, for example. So these organisms do have a bit of intraclonal variation, different classes of ribosomal RNA sequence. So that's one possibility we still need to sort out, whether this is some kind of sequence variation that we're picking up with our quantitative PCR. Alright, so another reason why doing some quantitative PCR is helpful for the monitoring programs is other problems discriminating particular species. So this is Alexandrium mathini, one of the organisms that I was using as the root for the phylogenetic tree. I know what the differences look like, but they can be really difficult to detect by routine monitoring. So if someone just showed me that picture and said, what do you think that is? I would probably say Alexandrium tamarense as well. Because the differences actually are only detectable by the position of a couple of pores right on the top of the cell. So if you can't easily image the top of the cell and the position of a particular hole in relation to other plates, then it's actually quite difficult. So routinely when you're counting fixed cells in a counting chamber, these would often be routinely referred to as Alexandrium tamarense. So even distinguishing some of the similar species is actually really difficult under routine monitoring. That's Austenfeldii there. Again, somebody who knows what they're looking for would probably pick the difference based on cell shape. If you're not overly experienced and familiar with this group of organisms, you could easily call most of those Alexandrium tamarense as well. You can see that dark sort of shadow in the middle of the top half of the cell. That's the sort of characteristic of Austenfeldii, this pore on the front of the cell that's much larger and in a different position. But otherwise, fair enough, I could see how you could easily call that one Alexandrium tamarense if all you had was a few cells in a counting chamber. Another one, Alexandrium margaleffi. I could pick that one quite easily. It's a species that has been around for a while. Shape, certainly I can pick it, but again, if you're not overly experienced at working with the species in this group, you could easily decide that it was Alexandrium tamarense. We also had a couple of examples where this organism was misidentified by the routine monitoring. It's not even in the same genus. It's actually a thing called Goniolax. Alive, in a live sample, I don't think you would confuse it with those at all. It's actually quite different. But when you fix the cell...
Those top ones blue because they're bioluminescent?
Yes, no, actually they're blue because I've stained them with a stain that shows the shell. So it's actually quite difficult to see the features of the shell in a normal light micrograph. It's because you get all this cell information. So they've actually been stained with a thing called calcifluor, which stains material that's structurally similar to cellulose, and the shell is quite similar to cellulose, and so you can look at them under a fluorescent microscope, which then will show us the shell without all the confusing background information from the insides. This organism, Goniolax hyalina, when it's fixed, I can kind of see how you could accidentally count that as Alexandrium tamarense. But to be honest, the shape is quite different. But again, it shows that under routine cell counting analysis that the biotoxin monitoring programs usually undertake, it's not so easy to work out what species is which when the cell is fixed. I don't think anyone would get this wrong if it was alive, but when you fix it with, in this case, a compound called Lugol's, which is an iodine, you lose any intracellular information, the shape of the cell changes a little bit, and you could easily get it wrong. So this is where incorporating quantitative PCR to just specifically detect and quantify the 3 or 4 problem species we have can make a big difference to the way we detect the organisms that cause these toxin problems. All right, so for those of you interested in further study, I've got a few current projects that are ongoing related to this. The temporal, spatial and temporal distribution studies I'm still doing using molecular detection approaches. We're reaching the end of a process of developing and validating qPCR-based assays for those different species. The other activity I've had going for a while is developing a sort of lab-on-the-chip instrument for detecting saxitoxins. So any research in this area, there's a few different options for those of you who are interested in research projects in the future. Okay, now you can ask me some questions.